We left off somewhere around here. We talked about tubular secretion, reabsorption, moving stuff from the nephron into the bloodstream, vice versa, the mechanisms that will control that and that kind of thing. One thing I want to mention now is something else that goes on in this area is the release of a hormone known as renin. We know that renal has to do with kidneys and all that kind of stuff, so this is where it kind of gets its name. But it's going to set into play a series of biochemical events that is going to help control your blood pressure. So the kidneys, there's a couple different, one of the things the kidneys do, I guess, is to help control your blood pressure as opposed to just filter waste and that kind of stuff. And if we can control where sodium goes, we can control where water goes. And if we can control, for example, more water coming into our blood vessels, we can increase the blood volume and thereby increase the blood pressure. So this is one thing that we can do to help control blood pressure. So what you're going to see is that a drop in blood pressure is going to produce a baroreceptor response. Remember, those are the things that sense pressure. Okay, so if these pressure sensors realize that the blood pressure has dropped a little bit, you're going to get an increase in sympathetic nerve firing, and it's going to signal those cells that actually can produce renin. So instead of reading all that to you, I want to show you this picture, and then we're going to go through it this way. If you take a look, renin is produced by the kidney, like I say, but it's actually produced by cells. It says up here, granular cells of the juxtaglomerular apparatus. Well, that's big words. What does that mean? Juxta means next to. Glomerular, remember the glomerulus, that little tuft of capillaries that was inside the Bowman's capsule? These are some specialized cells that are sort of in between the Bowman's capsule and in between those capillaries. They're sort of just part of the connective tissue, and they line that area. They're called granular cells, and they have the ability to secrete this hormone. So they can be induced by sympathetic stimulation. There can be a nerve firing towards them that can cause them to release their product. When they do, they release renin. Now, what happens is, as you can see, there's a lot of steps here. But the bottom line is, you have a protein called angiotensin. The key here is gen means the beginning of, the making of, the start of. Okay, so this is a precursor. This is something that exists that doesn't do anything as it is floating around right now. It does not act and turn into anything until it gets activated. And what you will see is that once renin becomes secreted, it can get into the bloodstream and come in contact with this stuff that's already made by the liver, this angiotensin. And when it does, these two things come together to make this product, angiotensin I. Now, what's next? Angiotensin I itself doesn't really do anything. It continues to float around. And what will happen is it will continue to go around your body until it comes to your lungs. When it gets inside your lungs, it comes in contact with cells that produce what's called angiotensin-converting enzyme, also called ACE for short. It takes this angiotensin I and converts it. So see, there's a lot of steps that are involved here. This was already there. Renin converts that. You get this. As this floats around, the enzyme that's produced in your lungs, by the cells in your lungs, can come in contact with that now. This previously was not here before renin was there. Now once that is there, these two come together, you get angiotensin II, the second step of this molecule here. Now, what does that do? That does several things. Realize the point of all of this is going to be to increase your blood pressure. Okay? Now, baroreceptors had sensed that the pressure was a little bit low. That's why this got released. Now, one of the things that a couple of things that can happen to help increase your blood pressure is to get more water into the bloodstream. How can you do that? Look down here. What angiotensin II can do is initiate the thirst response. It'll make you thirsty. It'll make you put more water in your body. It will cause the release of vasopressin. Vasopressin is also called ADH. It stands for anti-diuretic hormone. Maybe you've heard of diuretics. Diuretics make you release fluid. Okay? It increases urination. Anti-diuretic does just the opposite. It decreases urination. It causes your body to retain water. So vasopressin will do this. It'll cause your kidneys to absorb more water and release less water. So it puts it back in the bloodstream. So we put water back in the bloodstream through vasopressin, causing the kidneys to reabsorb more. You put more water in because it makes you thirstier. And at the same time, it's going to cause arteriolar vasoconstriction. It's going to make your arterioles constrict more. That's going to increase the blood pressure. All these things are going to work together to increase the blood pressure. Now, at the same time, what happens is, as some of this is still left over in the bloodstream and as it's moving around, it'll actually make its way to your adrenal glands, which are right on top of your kidneys. It's interesting because we started right next to them. We started with the kidneys right here, and the adrenal glands are sitting like right there. But instead of getting released and going straight to the adrenal cortex, which would save a lot of time, it actually has to make this whole path through the body before it gets there. It's a better control system. It's a better way for our body to monitor the in-between steps and exert more control over what's going on. Okay, so once it gets to the kidney, though, the adrenal gland, what it will do is cause the adrenal glands to release a hormone known as aldosterone. Okay, aldosterone then goes right back into the kidney, and that will affect how the kidney absorbs sodium. It'll actually cause the kidney to pull back more sodium into the bloodstream, and as a result, water will follow, and that'll help increase the blood pressure. So all of this is working together to increase blood pressure. And when it does increase, realize what happens. If all this causes an increase in blood pressure, the baroreceptors that started all of this are going to stop firing. They're going to stop sending a signal that the blood pressure is too low. All this will slow down. Renin will stop being produced, and as a result, all of this will slow down, and you won't have to try to increase blood pressure anymore. Now, what you can also see here is a lot of places where blood pressure control can go wrong. Why certain people have high blood pressure, certain people don't. There's a lot of factors. When you're dealing with enzymes and all these proteins, realize there's a genetic code that codes for all of these. You could certainly be genetically predisposed to having high blood pressure if any of these are not functioning properly, or if they're malformed whenever they get cranked out of the cells, that kind of stuff. Any of these pathways in between, something could go wrong from converting from this to this. Who knows? There's a lot of different ways. That's why there's this no one magic cure. There's no something that we can't go out there and just cure high blood pressure, because there's too many things that could go wrong with it. What about arteriolar vasoconstriction? We know that occlusion, diet, health, exercise, habits, all these things could help constrict arterioles as well. They could induce atherosclerosis and things like that. That's obviously going to have an impact on high blood pressure. So there's way too many variables involved here to just get a quick fix. Okay. Moving on, I want to mention congestive heart failure. I mentioned before when we talked about the cardiac unit that congestive heart failure is sort of a generic term used to say that the heart is not pumping out as much blood as it needs to serve the conditions. Now, who knows what all could cause that? There's a lot of things. So a failing heart would produce a reduced amount of cardiac output and a lower blood pressure from the heart. It's not pumping out as much as it should, despite an expanded plasma volume. Because what's going to happen is, realize, bottom line is, when you are in the early phases of, let's say, congestive heart failure, you're starting to get to the point where your heart is not cranking out enough blood and at a high enough pressure to meet the demands of the body. If that's the case, what ends up happening is your body realizes that the blood pressure needs to be higher because this blood pressure is dropping. But what your body doesn't realize is that kicking in all of the mechanisms that we just talked about to increase blood pressure, that's not really going to help. It's only going to make things worse. And I'll show you why. But realize that low blood pressure is not necessarily the problem. It's just a symptom of congestive heart failure. So what your body tries to do then is end up trying to treat that symptom, and it doesn't help the problem at all. I'll show you. But the triggered salt and fluid retaining reflexes are inappropriate. What I just said was you could cause sodium to come back into the bloodstream and water would follow. You could make yourself thirsty. You could drink more. You could release more vasopressin in your body, cause your body to retain more water. All of these things are true, but realize what's happening. If you continue to retain more water in your bloodstream and continue to try to increase your blood pressure in that way, the heart is still failing. The heart is trying to constantly push against the changes that you are making, and the reason that the blood pressure is so low is because it's trying to overcome it. It can't overcome it, and it's too low. It's not fighting hard enough against the backward pressure in the arteries. So by increasing that blood pressure, what you're doing is making it even harder for the heart to work. And whenever you make it even harder for the heart to work, It just makes it worse. It exacerbates the condition. And what happens is it's a feed-forward mechanism. Our body is trying to use what it normally uses to fix this blood pressure problem. But the problem is not just blood pressure, it's with the heart as well. So it ends up screwing up the heart, and it ends up making it faster in terms of getting worse. Okay, so what you get is an expansion of the extracellular fluid because you've got more pressure now. You've increased the pressure. You've got more fluid leaving the blood supply, going out into the tissues. You've got a higher level of ECF, more fluid in the ECF. This produces edema. It exacerbates or exaggerates or makes worse the failure that's already happening because the weakened heart cannot pump the additional plasma volume. You've cranked up more plasma volume to increase this pressure. The heart can't deal with it. So what does it do? It starts to retain it itself. You get more probably water like in the pericardial fluid. You get more backup in the heart. You get a sort of a swelling. You can get a hypertrophy of the heart and all kinds of stuff that could go wrong with this. So, okay. I already mentioned this. I explained this on the picture. Realize that everything will happen in reverse. If the blood pressure is too low, everything happened that we just said. If the blood pressure is too high, all the arrows point the other way, and everything can go in reverse. That's the point of this here. Okay. Now, the way that drugs work in terms of treating like high blood pressure and water retention and that kind of stuff, realize that it says some drugs affect reabsorption. I'm going to show you here in a second with how the kidneys actually work in terms of pulling in sodium, pulling in water, and that kind of stuff. There's a lot of different places. There's protein channels. There's enzymes involved. There's a lot of ways that you can use drugs to target these pathways. Diuretics are going to produce diuresis, which is increased urinary output. Kind of looks like diarrhea, which is increased output of the other stuff. Diuresis is actually increased urinary output. Okay, so diuretics cause that to happen. We can administer these things to help lower blood pressure. That's one thing that you can do. Get some water out of the body, and affect the blood plasma volume, decrease it that way. You could see ACE. Remember that stands for angiotensin converting enzyme. That's up in the lungs. That is a necessary component to help increase blood pressure. If we can make that not work and make that not happen, we can inhibit it, then we can help treat certain kinds of hypertension as well. So some drugs will target that enzyme right there, that angiotensin converting enzyme. If we target that, pull that out of the equation, we don't get as much, we don't get as high blood pressure result. We don't get as much plasma volume, that kind of thing. I also mentioned here ANP, which stands for atrial natriuretic peptide. It's probably in your book. It's atrial natriuretic peptide. It's very small. It's not considered a hormone because it's so small in terms of being a chemical. It is actually released by the atria, some certain cells in the atria of your heart, in response to pressure. Okay? If the pressure gets too high, what this will do is cause these cells to release this ANP. That will help your body inhibit sodium reabsorption. It will keep it from pulling more sodium out of the blood, or out from the tubes, and putting it back into the blood that will end up causing a higher blood pressure. Okay, now, as far as all the other stuff that's absorbed, we've focused on sodium and potassium, but there's a lot of stuff that our kidneys pulls in and out of the blood. Here's a couple, glucose and amino acids. We know that if we eat them, we put them in our body, our kidney has to deal with them. Kidneys have to deal with them. Now, I mentioned that they are going to be linked to sodium. We spend a ton of our energy on that sodium-potassium pump, stuffing that closet full of sodium so that they can diffuse and that kind of stuff. So to make this more efficient, what our body has developed is a way to link together the movement of sodium with the movement of these guys right here. They're going to end up hitching a free ride, basically, based upon how these things move around, what's moving where. These guys can get transported through what's called secondary active transport. It requires energy, but the energy is being burnt to move sodium, and these guys just hitch along. Okay? So you're going to find that this is going to happen in the proximal tubule. Remember where that is. That's up close. That's the first part. All right, now, one thing about all this stuff that's being absorbed is that with the exception of sodium, our body can move as much sodium as it needs to in and out of those tubules, okay? As much as it possibly needs to. But what you will find is that these things like amino acids and glucose, there's what's called a tubular maximum. It can only move so much at any given time, and whatever it can't hold and whatever it can't carry across that membrane, it's going to spill over into the urine, okay? So there's carrier molecules, like for glucose and for these other things, that actually have to physically hold them and carry them across this membrane. Okay? All right. In the case of sodium, if something is carrying sodium, helping carry like those sodium-potassium pumps, okay, that will be something that's carrying sodium from one side to the other. If they are full, if there's too much sodium and all those spaces are occupied, instead of just stopping, and instead of your body getting backed up at that point, aldosterone, that hormone that's going to contribute to blood pressure, okay, and sodium retention, it also causes more carriers to be made. So the cells that are in this tubule actually can make more of these carriers and compensate for the extra presence of the extra need to move sodium across there. So the point of all this is certain things have a maximum. They have a threshold. You can only transport so much at any given time. Sodium, there is no limit. Your body will respond and you will be able to transfer as much sodium as needed either way across that membrane. Okay. Too fast. Go back one. Now, fine. A thing to mention about glucose is that it is not regulated by your kidneys. It's regulated somewhere else. When we get into digestion, we'll talk about glucose and the liver and all that kind of stuff. But basically what your kidneys do is pull out all the glucose that's not supposed to be there. It's pulled out as much as it can. It is not regulated. It's just reabsorbed, and it's supposed to be reabsorbed 100%. You'll be able to pull out all the glucose that it spills over into the nephrons. Okay. Now, the filtered load of a substance is how much of that is filtered per minute. So, for example, with glucose, we actually filter about 125 milligrams of glucose per minute. So that's a reasonable amount. What's worth noting is its tubular maximum. That's 375 milligrams. That means that's the most that you could ever transport at any given time, given the carriers that are present that could move glucose for you. Those things have to be present, like I said, to cross that membrane. If you have more at any given time, if you have a higher level than 375 milligrams, trying to cross, realize that it will not get transported, it not get pulled back out of the nephron and put back into the bloodstream. What will happen is it will stay in the urine. And that's what we call spilling over into the urine. And it will stay there and get excreted through the body. And you'll be able to detect sugars. You'll be able to detect glucose in the urine at that point. Now realize the relationship there between that and diabetes, right? In the case of a person with diabetes, we know that glucose is not getting into the cell for some reason or another. So what happens is blood glucose levels spike. They get really high. They can actually exceed this amount whenever it starts to get filtered through the kidneys. If a person has too much glucose present in the bloodstream, its tubular maximum could be met, and it's pulling out as much as it can, but it's not pulling out all of the glucose present because there's not enough carrier molecules to grab them all. And our body does not just synthesize more like it does for sodium. You're stuck with that. Whatever you can't carry spills over into the urine. So there's the relationship there. Okay. So the point right at which it's getting saturated, the carriers are getting saturated. It's holding as much. These little guys are holding as much of whatever compound they're supposed to hold, as much as they can. We've reached what's called renal threshold. It's that teetering point where you can't hold anymore. Okay. Okay. Now, what your kidneys can do, I mentioned this here. We cannot control. We do not regulate glucose. But we do try to pull out as much of it out as we can, just to get it out of there. We let another body part regulate the glucose. We just pull it out. Over here, though, what you will find is that calcium and phosphate are, in fact, regulated by the kidneys. It can control how much calcium it pulls in and lets go. And what you will find, and you'll learn this even in your nursing classes, is that calcium and phosphate are inversely related. One of them, if one of those levels rise, the other level drops. Okay, they have to go in opposite directions. Like sodium and potassium sort of go in opposite directions. Calcium and phosphate are going to go in opposite directions. We'll hopefully learn a little bit more about that when we get into hormones and that kind of thing. But These are not hormones, of course. These are just chemicals. These are ions. But they are actively reabsorbed. We actually use energy to help control that process, where they are. Was there more in the ECF, more in the ICF, that kind of thing. Your kidneys will monitor that, and it is regulated. We'll talk about what they're used for and that kind of stuff later. Obviously, a big one is bone deposition. Calcium and phosphate are two of the biggest components of your bone tissue. But calcium, we know that calcium has to be used also like in heart contractions. We talked about the calcium plateau and how it's the channels that are used. And when we get into muscle contraction, remember the troponin, tropomyosin thing? Calcium had to move that rope back and get those things to work. So calcium is very important. We've got to have a way to regulate it. If we have too much of it, there's a big problem because we can't pull it out. We get constant muscle contraction. Everything sort of freezes up. You can certainly have negative impacts from that. We've got to be able to pull it back out of those spaces, so we can't have those levels too high. We also can't have them too low, or we will not get sufficient muscle contraction. So there's a certain balance that has to be achieved. The parathyroid hormone, when we get to endocrinology, we'll talk about that. But the parathyroid, right around your thyroid, it can change this. It can communicate with your kidneys and help your kidneys regulate based upon the feeds that the parathyroid gets in terms of how much of these things you need, what you need to increase, what you need to decrease. Okay, now, like I said, there's a lot of things that are moving just because sodium is moving. Okay? First of all, wherever sodium goes, number one, water is going to go. It's going to follow because of the osmotic gradient that gets set up. Number two, wherever sodium goes, chloride ions are going to follow because they are negatively charged and attracted to the positively charged sodium. So they're just going to kind of follow wherever they go. Now, 80% it says of water that gets reabsorbed, meaning pulled out of the tube back into the bloodstream. 80% of that water is obligatory, meaning that it has to happen, and it does happen, in the proximal tubules and the loop of Henle. Anything else is under finer control past that loop of Henle, like in the distal tubule and in the collecting duct and that kind of stuff. The remaining 20% is subject to hormonal control in the distal parts of the tubule, the distal tubule, the collecting duct. This allows us a finer, sort of more fine-tuned control by using hormones. It senses the... Somebody trying to get in? It senses the need. It senses from other parts of the body where there needs to be an increase in pressure, whatever. The hormones can be released in response to that. They can communicate with the distal parts of the nephron, and you can use other bodily inputs to help control that water there. Okay, I talked about osmosis. We know that there is a buildup of pressure here that's pushing things. Blood pressures help pushing stuff into here. There's a buildup of hydrostatic pressure that's sort of forcing stuff back. We've also got an osmotic gradient. These things become more concentrated in here, these plasma proteins, so naturally water is drawn back in the other direction, and when we factor it all out, we said there's about a net of 10 millimeters of mercury going in here that helps drive this and get stuff out of the bloodstream. Okay, now, another thing that happens is, I don't know if you remember this or not from basic A&P, but urea is actually not really part of the urine release. Urea is actually sort of travels. You've heard of like the BUN or BUN levels, blood urea nitrogen levels, that kind of stuff. It actually gets traveled through, it gets put back into your blood, travels through, and is degraded through other bodily processes and broken down. Gout, for example, is one that starts to crystallize, in joints and that kind of stuff. But anyways, the loss of water from the proximal tube, what happens is it does get filtered, okay? But it says the loss of water from the proximal tubule up in here, water getting pulled out. See right here's sodium can pass through here. It's going to end up getting pumped back out. The movement of sodium, water is going to follow. So there is a lot of water. We said a ton of water, right? 99% of the water actually gets pulled back out of here. Okay? And when it does, it increases the concentration of urea that happened to have got filtered. What will happen then is you'll find that urea starts to actually diffuse out. Because it's highly concentrated here, well, I guess the point of this is to show you that for every reaction you see there's going to be another reaction that is directly related to it. The fact that sodium-potassium pumps are moving out sodium means that you get a free ride with water. Water's going to follow. You'll also find that glucose can follow. You'll also find that by removing water, you end up leaving more urea behind. When you end up leaving more urea behind, you set up a diffusion gradient, and it starts to get concentrated and then starts to leave on its own because it's a higher concentration in here than it is out here. So it's sort of a feed. It's all connected. It's all can be traced back to this initial sodium movement. One thing leads to another. Before long, everything can start moving where it needs to. Okay? So urea then can get pulled back out and put back into the bloodstream because it gets concentrated in here. All right. Now, I mentioned that we've been talking about pulling stuff out, tubular reabsorption. Now let's look at secretion. We know that sodium gets pulled out, it gets cranked out here, loaded up in here, and it diffuses into the bloodstream. Okay? Potassium is going to do just the opposite. Potassium is going to diffuse out here. It's going to have to get forced in here so that it can diffuse out into here, okay, in the opposite direction of sodium. Okay, so potassium ions are going to be secreted, whereas sodium ions are mostly reabsorbed. Okay? Hydrogen ions are also going to be secreted. Why would hydrogen ions be secreted out here? What did I say? Do you remember what hydrogen ions are associated with? Okay, acidity, right. Acidic conditions, acidosis. One of the things that you'll find that our body obviously is able to do is to maintain pH homeostasis. There's a lot of ways we do that. Breathing. We get rid of carbon dioxide that can contribute to acidosis. Another thing that was going to happen is if it makes down here, if hydrogen levels are high in the blood, what our kidneys can do is pull out some of that hydrogen and put it in the urine. Okay, get it out of the body. And so by doing that. So hydrogen and potassium can get pulled out of the blood and put back in here into the waste. There's other organic ions. These are larger things, larger products that couldn't have got filtered before coming through. Okay. Now, I say potassium secretion is controlled by aldosterone. Why? It's because sodium reabsorption is controlled by aldosterone. I mentioned a while ago that aldosterone is a hormone that causes your body to retain sodium. It causes your sodium to be pulled out of the nephron and put into the blood. So if sodium gets caused to pull out, then what that means is potassium is going to go the other way. It's going to get put in. So that's the link with these. Okay, the pump as far as what's happening at this basolateral membrane is the same as it was with sodium. It's just in the opposite direction. It's the same pump. It's just instead of pumping out, we're looking at pumping potassium in and cramming up the potassium in here so that it diffuses out. With sodium, we were letting it naturally diffuse from here into here, cranking out the pump to load it up out here so that it would diffuse into the bloodstream. Here we're working backwards. We have a lot of it here, it diffuses into here, and then we've got to crank it and cram it into here so that it will flow out into there for potassium. Yeah, diffusion's passive, but this pump is active to cram it. Right? Okay. All right. Let's see, what we got here. Oh, so this is, like I said, this is a little bit redundant, but this is the same thing about opposite directions. If you're going to promote, aldosterone is going to promote sodium reabsorption, pull it back in at the same time, promote potassium secretion out. They've just got to balance each other out. That's all that's got to happen. Okay. We already know now, based upon what we've looked at with Chapter 4 with neuronal physiology, what we looked at with muscle physiology and cardiac physiology, we know the importance of potassium, sodium, calcium into just those three, let alone other parts of the body, how it works. So, especially for membrane excitability, depolarization, we've got to have proper levels of that stuff. So it's a very, very, very important job that our kidneys are able to maintain all of this. Okay, okay. I mentioned organic anions and cations. These are just charged particles, negatively charged, positively charged particles. Organic, these are going to be things that you get in. Bigger molecules, like from drugs that you take, additives that we use, all that kind of stuff that sort of doesn't get dealt with under natural situations. Well, I mean, in terms of like specialized pumps for it and that kind of stuff. This is just what's left over here. You'll find that in order to eliminate this, they don't make it, especially because of their size. They tend to be bigger molecules. They don't make it through that filtration step at the very beginning. They don't get pushed through that glomerulus and get into the Bowman's capsule. Instead, they sort of stay in the blood, and they stay in that little mesh of blood that's all around the nephron. So if you want to get rid of them, what we have to do is be able to secrete them from the blood back into those tubes, and that's what we're talking about here. Okay? So if it wasn't for that, if it wasn't for our body's ability to do that, realize that that would stay in our blood and obviously create a toxic type of situation. So we've got, for example, food additives, drugs, epinephrine, even histamine that's released during an immune response. Those would all be things that have to be, they cannot be filtered. They have to be pulled back out of the blood physically into the nephron through tubular secretion. All right. As far as urine goes, I might have mentioned that already, but you get about 1 milliliter on average of urine per minute. Remember we said there was 125 milliliters of filtrate that was produced, and 124 milliliters of it actually gets reabsorbed by the time it's all done. So we end up with only about 1 mil a minute. That's an average, of course. So about 1.5 liters are excreted every day, again, average, out of the 180 liters per day that are filtered. Like I said, that would make for a much more interesting day if we actually had to excrete 180 liters per day. Just pretty much. We could just wear NASA diapers. Super absorbent. So. All right. Now, what I want to point out here, though, is that it only takes a very, very, very tiny, small amount of a change in terms of how much filtrate is reabsorbed. Even if they're just a tiny amount more than this, than what we're talking about up here, that's reabsorbed or not reabsorbed, that can make a huge change in terms of how much urine actually forms. And what I'm talking about is, like, for example, if you're used to pulling out a certain amount of one of these things from the bloodstream and putting it back into the nephron, or vice versa, you're used to pulling it out of the nephron and pulling it back into the bloodstream. All it takes is a very, very small, tiny difference, either more or less, of that movement to produce a huge difference in the amount of urine that's produced. Okay? So when you get a large amount of difference in terms of urine production in the body, realize it's probably due to just the regulation, a small change in terms of what is being moved across the kidney. And I'll show you how that's possible here in a second. All right. Now, I'm going to mention something called plasma clearance. Instead of reading all this, let's look at just an example. Plasma clearance is actually, it's not that bad to understand if you consider what we got up here. We know that filtration is this process. And what happens is when it gets pushed through, what this rectangle represents is just a given volume of filtrate. Okay? And within that volume, there's this much particles in there. Okay. The glomerular filtration rate is how much filtrate basically is produced in a given amount of time. That's the glomerular filtration rate. Now, what's the difference between that and the plasma clearance rate? It's a little bit, they're very similar, but slightly different. Notice that the plasma clearance rate is, instead of looking at what forms in here, we're looking at what disappears out here. Okay? So what you will see is how much of your plasma has been cleared in terms of whatever it is we're looking at here, whatever these purple dots represent. How much of your plasma has been cleared, and at what rate is it being cleared? So, for example, if something is filtered, and whatever these dots represent, if it is filtered and it becomes part of this filtrate, okay? If it is filtered but none of it gets pulled back out through tubular reabsorption, no additional amount of it gets secreted back in by the bloodstream, then whatever you've pulled out is what stays out over here. Okay? So we're going to say that the glomerular filtration rate is equal to the plasma clearance rate in this particular example. However much you pull out stays that way over here. I'll show you the difference when we get a different scenario. Take a look at what's going on over here. What if you get this same filtration, so you've cleared, here's how much, but what happens is none of it got secreted back in, right? The blood didn't put any more of it back in, otherwise you would have taken the number up. What happened, though, was your blood reabsorbed it, okay? It actually pulled, even though it got filtered, it pulled it all the way out. For example, like you would get with glucose. Glucose was filtered, right? But but it's 100% reabsorbed. So you may have a glomerular filtration rate for glucose because it actually gets filtered. But how much of your plasma actually got cleared of glucose when it was all said and done? None of it. You didn't clear anything. You didn't clear any glucose from the plasma. It actually got put back in. Now, for a second, you could say that maybe there was a little bit of a clearance right there, but I'm talking about net when you're done. Overall, what happened? Your glomerular filtration rate was represented by this, but it eventually got reabsorbed, right? So your plasma clearance rate, like in this case, would be 0. You didn't clear any of it from your plasma, even though you filtered some of it. So in this case, we'd say the plasma clearance rate is actually less, much less, as given here, than the glomerular filtration rate. Okay, so that's what that means. Take a different example. What if you get a filtered situation like this, but none of it is pulled back into the bloodstream. Instead, some of it actually is additionally pulled out of the bloodstream. And you've pulled even more into the tubule, into the nephron. Okay? Even more tubular secretion from the blood into the nephron. So how does your plasma clearance rate compare to your glomerular filtration rate? You have actually cleared, since some of it is actually leaving and going back in, you've actually cleared from your plasma more than you originally filtered. Right? You see how that works? Over here, you cleared from your plasma basically nothing. Much, much less than what you actually filtered because it was ended up being all put back in. So here, your glomerular filtration rate is much greater than, okay, your glomerular filtration rate is much greater than this actual plasma clearance rate. Your plasma clearance was virtually 0. Over here, your rate is given here, but look, you've actually pulled even more back in. So your actual plasma clearance rate is greater than just what you filtered by itself, than your filtration rate. Okay? You'll have to go back and take a look at those and see if you can make sense of that. If not, obviously ask me. But, so to go back a couple slides, I wanted to mention just with this particular compound, it says if a substance is filtered but not reabsorbed or secreted, its plasma clearance rate equals the glomerular filtration rate. That's what I said. That corresponds to this right here. It's filtered, but it's not reabsorbed. Okay, it's not go from here to here. It's not secreted. It doesn't go back this way. So whatever you filter is equal to whatever you clear, basically. Okay? Now, I mention this because inulin, not insulin, there's a chemical called inulin, is an ideal substance that you can inject into the body to run a test to help compute a clearance rate for someone because it is not filtered. Sorry, it is filtered, but it is not reabsorbed and it is not secreted by our body. It's a product that's just is there. It stays there. Whatever gets filtered out, that's it. That's all you get. You don't get any more of it pulled out. You don't get any more of it put back in from the blood. So this is actually an ideal synthetic substance. Oh, it's not synthetic, but not necessarily in our body. A substance that can be used to compute a clearance rate for someone. You can keep track of how much you put in, keep track of how much comes out in the urine, and compare that. Creatinine is actually something inside of our body that is only filtered. It's a natural substance. There's just not that much of it in order to run a test on in our body. So. Okay. Now, I'm going to rely on a video here for this because this is a little bit abstract. What our body is able to do is to control the concentration of urine that we produce. And it's a very sophisticated way in which it does this. And it's designed, the mechanics is really cool in terms of how it's designed and how we've got specialized cells that have certain channels and other ones that don't, and they work so closely together to help control sodium movement and water movement to get these concentrations. So I want to show you this video. But what I want you to see is that here's your kidney, here's your cortex, here's your medulla down in here. These numbers represent osmolarity, in other words, a concentration. Basically, the higher the number, the more, like, let's say, salt is present. Okay, not entirely accurate, but close enough. So out here, you'll see farther away from this drainage site down here, Farther out here, there's a lower concentration. There's more water, less dissolved solutes, less concentrated. Down here, there's going to be less water, more things like sodium and chloride. How does this play out? How does our body do this? How does it control these? And how does that impact in the amounts and concentrations of urine that we form? Now, it's all going to be, all of this regulation that you're going to see is going to happen in, most importantly, in the loop of Henle. So we're going to look real closely at the loop of Henle and what is happening there. Okay? It's going to be, what's going to drive all of this is that it's just going to be basically two things, sodium and water. Are the cells permeable or impermeable to sodium and water at a given location? And if so, how is that going to affect how they are moving? So, let's take a look at this. Oops. Volume down.
Here's a simplified view of the cortex and medulla. This theory is called the countercurrent theory because of the opposing flow of filtrate within the two limbs of the loop. The complex interplay of the ascending and descending limbs forms and maintains an interstitial osmolarity with a gradient from approximately 1200 milliosmoles near the bottom of the loop to the normal 300 milliosmoles near the cortex. The gradient formed by this activity is essential for the concentration of urine. To demonstrate how the gradient is formed, we will look first at the role of the ascending limb of the loop and then at that of the descending limb, because the ascending limb creates the conditions necessary for the descending limb to function. Click the ascending limb to begin.
Okay, what we're going to look at here is, this is your loop of Henle, obviously not drawn exactly like it would look. But we're going to start, instead of starting over here, we're going to start to look at what happens over here, because it's all connected. We're going to look at how sodium and water is actually moved out of here and how you can end up with all these concentration gradients and why you should care about these concentration gradients and what happens if they get screwed up and that kind of stuff. So what we're looking at here is the ascending loop. We're headed towards the distal tubule. You can see down here in the real nephron what this would correspond to. Okay?
The ascending limb of the loop actively transports sodium chloride into the interstitium, increasing its concentration while restricting the diffusion of water. The interstitium surrounding the tubule becomes more concentrated, while the fluid inside becomes more dilute. As the fluid moves up the loop, there is progressively less solute to pump out, so the highest concentrations of solutes are near the bottom of the loop. Now click the descending limb to continue the process.
Okay, the key here is what you need to realize is the cells that actually make up the ascending part of the loop of Henle, they have protein channels on them that are of a certain type. They have the ability to actively pump out sodium into the environment, and it will do that. They'll keep pumping sodium out. And just realize, if you start down here, and as this fluid is climbing, as it's being pushed through, obviously you're going to have more sodium pumped out down here than you are up here because it's going to progressively get less as you go up, just like with anything else, right? If you had a leak in a hose, whatever, you're going to have less water at the end than you are at the point source, way down here. Okay, things sort of fan out. I use the analogy of when we talked about the electron transport chain. I talked about going to a mall the day after Thanksgiving, right? And everyone weeds out and everyone sort of dishes out and all the stores are taken, and then at the end there's progressively fewer people than there was to start with. The same concept. The sodium starts to dish out, and you'll see that those numbers, remember, were a lot higher at the bottom of this loop, like 1,200 as opposed to 300 at the top. All of this is permeable to sodium actively. It'll pump it out, but it just, because of the geographical location of this, where it's located, more of it gets pumped out towards the bottom and fewer of it is left to get pumped out at the top. At the same time, what's important to note is all of the cells that are in this ascending loop of Henle are impermeable to water. They will not let water follow that sodium. It actually closes down and prevents water from traveling through. Okay? And that's important because that's going to sort of create this little, you're sort of like how you can create a dam. You can create a dam and hold back water, and you can control when that water falls and all that kind of stuff. So sort of we're harnessing energy in a very creative way here. Okay? All right. Take a look at what's happening on the other side at the same time.
The role of the descending limb in this process is to provide a continuous concentrated supply of sodium chloride to the ascending limb. The filtrate concentrates as it moves down the descending limb because water diffuses into the higher osmolarity interstitium created by the ascending limb. The solutes remaining in the tube reach a concentration approximately 4 times higher than normal body fluid. The effect is much like what happens in the Great Salt Lake when water evaporates and salt is left behind. The fluid that is left is very salty. This salty filtrate provides a ready supply of sodium chloride for the ascending limb to maintain the osmotic gradient.
Okay, a couple points about this. Number one, we got...
Click the interstitium and watch the combined but asymmetric activities of the two limbs as they build the osmotic gradient.
What we've got here is sort of the same thing. Water is coming in. What we have is impermeable to sodium chloride. Sodium chloride cannot escape over here, just like water could not escape over here. So what you get is water progressively coming out more up here and less of it coming out down here, right? And what that ends up doing is concentrating the amounts of sodium chloride by the time you get down here, so that when it starts to climb, you've got a whole lot of sodium ions in here. They start to dish out whenever these things open up and allow them to, and then more of them come out here, less of them are available up here. So these two sides work together to create this and make it happen. Okay? Water naturally wants to leave because it's being drawn out. There's a higher solute concentration out here, drawing that water out, making it go where it needs to go. So water leaves over here, sodium can't. A whole bunch more water leaves up here, helps dilute that, make it less concentrated. Less water is available down here, allowing a more concentrated situation. All the while, sodium could never leave. Now over here it's able to leave. It's going to start to leave through active transport. So it's going to become more concentrated down here, less concentrated up there. That is in addition to and coupled with what just happened over here. So you get these layers where it's less concentrated up here, much more concentrated down here. Now the reason that all of this is even important is because if this did not happen, and this was not here, we could never produce any urine in our body that was more concentrated than what our body tissues are. And we know that concentrations of urine can get very concentrated, and they can far surpass concentration of solutes and other things that we find in our extracellular fluid. So, because what you got to realize is if we were just doing tubes and this little interplay wasn't happening, it would get a chance to balance itself out, water would move according to where the solutes are, and you would never be able to produce urine any more concentrated than just what our ECF was. This allows us to do that. By highly concentrating these areas, by playing in with this, we can get highly concentrated urine, or we can get far less concentrated urine even than what's in our normal ECF. Okay? How is it related to blood volume? Because the more, as you'll see up here, first of all, a lot of things can be affected right here. Hormones can come into play right here and cause more sodium to come in. The bottom line is blood volume is going to be according to how much sodium is actually left, how much is pulled out, how much is still in here versus how much is in the bloodstream. If there's more sodium still in the bloodstream, we increase the amount, we pull out less sodium, leave more sodium in the bloodstream, more water is going to follow, you're going to increase blood volume, you're going to increase blood pressure.
The way the two limbs cooperate to multiply the solute concentration from the normal 300 milliosmoles of the cortex
so watch as it gets stronger.
4 times higher in the medulla is called the countercurrent multiplier mechanism. This mechanism has also diluted the luminal fluid in the ascending limb to an osmolarity of approximately 100 milliosmoles. As a result, water is retained while solutes are removed. This graphic now shows the 200 milliosmole difference between filtrate and interstitium developed by the cells of the thick ascending limb.
200 they were talking about was right there, okay, at the very top of the ascending limb. Now, okay, now, that link's on there if you want to go back and take a look at that. But now let's look at everything together for sort of a total review.
The major excretory organ of mammals is the kidney. Humans have 2 kidneys kidneys located in the upper rear region of the abdominal cavity.
Some people have
The
three.
urine they produce is
Some people just
conducted
have
to
one
the
giant
urinary
one that's a
bladder through
horseshoe.
the ureters. The urethra drains the bladder. The internal structure of the kidney includes an outer cortex and an inner medulla. The ureter divides into branches, the ends of which envelop medullary tissues called renal pyramids.
I told you, right, that like the pictures you see in your textbook is only like represents like 70-some% of the actual population. So, yeah, there's a lot of people out there that are anatomy.
The actual work of the kidney is carried out by functional units called nephrons. Each human kidney contains about 1 million nephrons.
The guys are just working like dogs in there, and we just abuse them.
An afferent arteriole carries blood to a knot of capillaries called the glomerulus. Draining each glomerulus is an efferent arteriole that gives rise to the peritubular capillaries, most of which surround the cortical portions of the nephron tubules. Blood pressure forces water and small molecules to be filtered from the glomerulus and collected in Bowman's capsule. The initial segment of a renal tubule is called the proximal convoluted tubule. The glomerulus, Bowman's capsule, and proximal convoluted tubule of each nephron are located in the cortex. From the proximal convoluted tubule, the nephron tubule turns down into the medulla. The portion of the tubule in the medulla is called the loop of Henle. Where the ascending limb of the loop of Henle reaches the cortex, it becomes the distal convoluted tubule. The distal convoluted tubules of many nephrons join a common collecting duct in the cortex. The collecting ducts then run in parallel with the loops of Henle down through the medulla and empty into the ureter at the tips of the renal pyramids. A few peritubular capillaries run into the medulla in parallel with the loop of Henle and the collecting duct and form the vasa recta. These capillaries carry away the molecules that are resorbed from the tubules. All of the peritubular capillaries join back together into a venule that eventually leads to the renal vein. Nephrons regulate the composition of blood and urine by a combination of filtration, secretion, and resorption. Viewed schematically, we will see how these processes are facilitated by the regular arrangement of segments of the nephron. The proximal convoluted tubule is responsible for most of the resorption of water and solutes from the glomerular filtrate. The cells of this section of the nephron actively transport sodium ions and other solutes, such as glucose and amino acids, out of the tubule fluid. The active transport of solutes out of the proximal convoluted tubule into the tissue fluid causes water to flow by diffusion. The water and solutes moved into the tissue fluid are taken up by the peritubular capillaries and returned to the venous blood, leaving the kidney. Despite the large volume of water and solutes resorbed out of the proximal convoluted tubule, the overall concentration or osmolarity of the fluid that enters the loop of Henle is similar to that of the blood plasma, although its composition is quite different. The ability of the kidney to produce urine that is hypertonic to the blood plasma is due to the loop of Henle. The loop of Henle does not concentrate the urine directly. Rather, it functions as a countercurrent multiplier, creating a concentration gradient in the surrounding medulla. To understand the countercurrent multiplier mechanism, it's easiest to move backward through the tubule, starting with the thick ascending limb. The thick ascending limb actively transports chloride ions from the tubule fluid and moves it into the surrounding tissue fluid. Sodium ions follow passively. The thick ascending limb is not permeable to water, so the resorption of sodium and chloride ions out of this part of the tubule is not followed by the outward diffusion of water. This resorption of sodium and chloride ions raises the concentration of solutes in the surrounding tissue fluid. The descending limb, in contrast, is permeable to water, but not very permeable to sodium and chloride ions. Since the surrounding fluid has been made more concentrated, water leaves the tubule by osmosis. As a result, the fluid in the descending limb becomes more concentrated as it flows toward the bottom of the medulla. The thin ascending limb is not permeable to water. It is, however, permeable to sodium and chloride ions. Since the tubule fluid is more concentrated than the surrounding tissue, sodium and chloride ions diffuse out. the thick ascending limb continues to move sodium and chloride ions to the medulla by active transport. As a result of this process, the tubule fluid reaching the distal convoluted tubule is less concentrated than the blood plasma, and the solutes that have been left behind in the renal medulla have created a concentration gradient in the surrounding tissue fluid. Since the fluid entering the distal convoluted tubule is less concentrated than the surrounding cortex, the tubule loses water osmotically as it flows toward the collecting duct. The tubule fluid entering the collecting duct is at the same concentration as the blood plasma. However, since sodium and chloride ions have been moved out of the tubule fluid, urea and other waste products make up a greater proportion of its total solute content. As the collecting duct descends from the cortex to the tip of the renal pyramid, the concentration gradient established by the loop of Henle increases. This increasing solute concentration causes more and more water to be absorbed from the fluid, thus concentrating the urine in the collecting duct.
Seems like a long... Whoa. Hello? Seems like a long way to get there. There's a lot going on. But if it didn't work this way, it wouldn't be nearly as efficient. No, not here. It's impermeable. Well, only at the loop of Henle, which is down here. Yeah, it's definitely not. Because the bulk of sodium that is pulled back out actually happens right here, like a huge chunk of it. So no, it's not impermeable there. It's certainly where the loop of Henle starts. Once you hit the medulla and the loop of Henle starts, that's when the impermeability starts, and it can't get out there. So what you see is by layering this like this, you can see, like in the color, they've chosen the nice color of yellow to represent our familiarity. Sort of more yellow here, the more concentrated it is. And the same applies for this collecting duct that's traveling alongside of it in parallel. So what this allows is, by sort of working this way and coming back and allowing things to move and in and out, it allows us to create situations where things can move from our blood into there. And more importantly, we retain a lot of water. We save a lot of water because as this collecting duct now, well, we're assuming that as we get here, a lot of the stuff's already been moved in and out, and it's actually just in the collection process. And what it does then is sort of at a final stage pulls out any extra water that it can. And it wouldn't be able to do that if it weren't for all this gradient that was set up because of everything that was happening over here. Okay? This was basically just a big setup, is what this was, so that all of this water can be pulled back out and we can more highly concentrate the urine that we are excreting so that we retain water and recycle it and use it for our bodily needs. So if it weren't for this, though, this could never happen. This whole gradient out here that's causing this water to leave would not be there if it weren't for what was happening down here in this loop of Henle. So it's all kind of working together. All right. Now, we can affect that at different points. Okay, we can go in there and knowing what's happening at these different parts of this process, different drugs can act differently, different hormones can act differently on certain places. I say vasopressin-controlled variable water reabsorption. Vasopressin is, like I said, ADH, which stands for antidiuretic hormone. It's going to cause you to retain water. The question is now how? How can it do that? What it will actually do is work there in those final tubular segments, that distal convoluted tubule, the collecting duct, down in that area where all this stuff was happening. If you can control the amount of that water that's being pulled out and do something to cause it to stay back in that area, you're going to force a whole lot more water to stay in, and that kind of thing. Or, vice versa. If you're trying to, in terms of antidiuretic, you want to retain water, then what you want to do is pull more water out of the nephron, right? Because realize whatever's in the nephron is going to go get it excreted. So if you want to anti-diuresis, if you want to minimize the volume of urine, the volume of water in there, you want to pull more water out of the urine so that it goes back into the body. So this is what can happen here. So It says 65% of the water is obligatory in the proximal tubule. That means it's just going to happen. Okay, there's not a whole lot of exerted control over it. 65% of that water is going to get sucked out just because there's too much of it there and there's too much to work with. And 15% in the loop of Henle. Now, where you're going to get the most variation and the more fine-tuned control over water is in actually the distal tubule and the collecting duct. These processes I just showed you, because of those concentration gradients, that's where a lot of that water gets to leave and where it starts to concentrate that urine. We can secrete ADH, which is also known as vasopressin. Okay, and what will happen is, notice what it does. Okay, the secretion of vasopressin, or antidiuretic hormone, increases the permeability of the tubule cells, okay, to water, which was formerly impermeable, right? We just said that on that distal part, especially at the top of the loop of Henle as we're coming out, it's impermeable to water on that side. If we mess with that and we make it more permeable to water, we're going to change a lot of things. Okay? We're going to allow more water to escape, and when more water escapes, we can even concentrate the urine further at that point, and we can retain more water back in the body and not have it stay in those nephron tubules. Okay? It says, look at the next slide, so we'll take a look. What will actually happen is here's the hormone coming through the bloodstream. It can take on to the basolateral membrane of these cells right here that make up the tubule. There's receptors there. You can get a link up. You get a second messenger gets kicked in, and what ends up happening is you affect these channels that were normally impermeable to water. Now they can open. So it's a hormone-induced response. Once these start opening, more water leaves the tubule. Okay? Remember, originally it was locked up in there. More water now can leave that tubule, and when it does, look where it's going. It's going to have to exit the tubule. The only place it really ends up getting to go is back into the bloodstream, okay, and back into the ECF, back into the body. Okay, so that's going to be a way to conserve it. Okay, just so you know, and I'm going to discuss this with the endocrine chapter, but vasopressin is actually produced in the hypothalamus, which is up here in the front part of your brain, sort of in the middle, I guess, and stored in the posterior pituitary. I'll explain all of that later. Okay. It also can signal the collecting duct, which facilitates the reabsorption of water. So we've got up here, we talked about just the tubule in general, but realize it can go to the collecting duct and actually cause the body, because of opening these channels, to bring more water out and put it back in the body. So I showed you this, cyclic AMP. It's the second messenger system by which it works. Whoops. It is an antidiuretic, so that means it puts water back in. It causes you to not put so much out. Okay. Let me see real quick. There's only got to be a couple slides left, so I want to go ahead and finish it. Okay. All right. So we about got it. Just stick with me here. It says, let's see. Let's see if I want to say anything here with this. Already talked about this. We already know that. Hold on a minute. Okay, I don't think that's nothing that we don't know already, based upon what we've talked about. So, all right, we know this obviously in terms of renal failure. The implications of this is really bad. I mean, obviously we have heart and brain, and that's bad. But as far as what can be affected by the most different types of things in the body and how it can have such far-reaching consequences, renal activity is a big one, and we've got to make sure that these things, because they can affect so many things. If we screw up our ion concentrations, everything gets screwed up. It could be our heart, our muscles, our brain, you name it. There's so many things that could go wrong with kidney failure. So it's of utmost importance. It could be an infection. An infection could get in there and destroy kidney tissue, in which case they're not going to work as well. Realize with tissue damage, you affect not only functionality of the cells, but the porousness and the filtration. If cells are destroyed and it's not filtering properly, you can get blood into the urine, bigger holes basically, through which to filter. Things can get through that are not supposed to. Plasma proteins, blood cells, you name it. All the bad stuff. Okay, toxic agents. Obviously, if you start to accumulate toxic agents into your kidneys, they're going to work on those cells. They're going to work on the destruction of the cells that make up those tubules, that make up those capillaries. You're going to be able to destroy the kidneys in that way. Inappropriate immune responses. People can have an autoimmune response to destroy a spillover and destroy some renal tissue and that kind of thing. Obviously, kidney stones or any kind of other obstruction of urine flow, you get a backup in that, you get a overconcentration. Worst case scenario, not only do you screw up osmotic gradients, but some of these things can precipitate and crystallize if they get to stay in the kidneys, which is sort of like where kidney stones come from to begin with. But other things, calcium oxalate crystals can start to form, other types of things that get highly concentrated in these tubules. If you get enough of it in there and you don't let it dilute like it should, it can actually recrystallize and form solids in there, in which case we've got a big problem. Okay? Obviously, blood supply. Insufficient renal blood supply could certainly stroke those tissues. This is easy. We know the bladder stores urine. The actual process, I don't know if you knew this or not, but there's a name's called micturition. So urination, micturition, it's the voiding process. We know the bladder stores urine. There's actually stretch receptors in the bladder that respond. Anywhere from 250 to 400 milliliters. Remember, like a beaker that we normally use, the beakers that we use are about this big, about that big around, looks like the size of a coffee cup. That's about 250 milliliters. So generally, if you get about that much in you start to feel the urge to void. It could be up to anywhere about 400 milliliters. So now you kick off this reflex. I think it becomes, I don't know, like 700 milliliters or something. It becomes distended to the point where, you've gone, well, you're going to do damage at this point. You've went past what normal distension would be. Okay, it's all nervous related though. So once this reflex, those stretch receptors kick in, muscles start relaxing. Actually, bladder muscles start to contract. Urethral sphincter muscles, at least the internal urethral sphincter muscles, start to relax, and then you get more of a pressing feeling that you feel. So the contraction of the bladder is going to push that out. I mentioned here the external versus the internal urethral sphincters. Realize that one is voluntarily controlled, one is not. The first one the first part is sort of like pinching off the urethra. Okay, the first one that happens is not much under your control. Okay, it's going to kick in this reflex. It's going to let go. It's going to increase the pressure, and it's going to increase the urge to void. The second one is more under control. That's how come you can hold it in. There's a lot of things that can affect this, obviously. Pregnancy is a big one. That'll affect how the response of these sphincter muscles and that kind of thing. All right, I don't need to know all this. We know that I've already heard it all, but we know that incontinence is the inability to sort of prevent unwanted relaxation and discharge and that kind of thing. So now that could be based upon just the sphincter muscles themselves having been stretched or whatever, compressed, the whole pregnancy thing, or it could actually be a nerve-related type of situation, spinal cord injury, nerve damage, whatever that controls these muscles. So, all right, that's it.